Namaste to India, bonjour à la France, buongiorno, Italia, buenos dias to Spain and our friends in Latin America. Good morning and good afternoon to those in the US and on the East Coast, and good evening to those in UK. Oh, now we have a Swiss watch too. Well, I have to speak several languages too to say, okay, so let's do this. Let's do this. Well, good morning again, and good afternoon to those on the East Coast. I'm Nathalie Cabrol, and I'm a planetary scientist and an astrobiologist. I wanted to thank Sanj for inviting me to share some thoughts about our beautiful discipline today. So I guess I'm going to start to tell you a little bit about who I am and how I came to be an astrobiologist. Today I am wearing two hats. One of these hats is being a scientist, which I am, very much involved in science and projects and exploration. And the other one is being the director of the Carl Sagan Center at the SETI Institute, which is in itself a wonderful exploration and adventure, and also an important part of this aspect of being a scientist. I'll come back to that later. But to get started, I'd say, how I came to become an astrobiologist, I guess, I don't know. It was always in me. As back as I can remember, I wanted to be not an astrobiologist because the term didn't exist at the time, or even planetary sciences. The term really didn't exist at the time. I was born at the time when Armstrong stepped on the moon, and so for me, like everybody in this generation, that was this aha moment. I knew I wanted to be somebody who would be doing something related to the universe and understanding the stars and the planets, but I couldn't phrase it very well. I was very little. But when I saw Armstrong, I said, this is what I want to do. And the vision of Armstrong and the logo of NASA all of a sudden became one big goal in my life. And so I really applied myself from the very beginning with that goal in mind. And this is so important for all of you who are looking to, what you want to be and what you want to do, whatever you want to do. And this is one thing that my mom always told me. No matter what you want to do in life, whether it's selling shoes or being an astronomer, being a seller, being a baker, apply yourself to it and become the best at it, because this is all we have, the passion we have for life. So I applied myself to it, and I was a straight A student in pretty much everything except for one thing, and that was math. And this is not really a good news when you want to be an astronomer. So mathematics and physics came late to me. In fact, I was more of a hands-on person, which means that I started to understand math and physics when I was in my late 20s, and I started doing experiments on my own. Then it made sense. Before that, I just didn't get it. But going back in time, it was clear that a astronomy path for me would not be straightforward. And there was a heartbreak right there, but at the same time, you cannot stay and live on heartbreaks. You have to move on. And something I knew really was something I enjoy was a naturalist side of myself, which means that I loved being out. I loved to be understanding processes and looking at nature, understand it. So I went to the university, one of the Paris universities, since I grew up in France, and started in earth sciences. And then I really loved it. I really loved it because there I could understand one planet, and that was ours. We often forget that, yeah, we are on the planet too. And I started to understand what geomorphology was, what natural hazards could mean, and I learned remote sensing, climate, hydrology, a number of things, and mapping that would come very, very important later on as I was moving on to planetary sciences. But at the time, I was very focused on earth sciences. I was very happy at the University of Nanterre where I was, and ready to actually start a master in environmental hazard. And that was perfect because my lab was actually doing some studies for the French Ministry of Environment. So there was an application to what we were doing, and we were out all the time. We were looking at avalanche corridors, at floodings, at all sorts of things. I was happy there. I was really happy. But then, of course, I had not forgotten about my dreams of astronomy, but I had put it on the side and said, well, I'll be an amateur astronomer. But I was still having these magazines with me and always talking about astronomy. And one day, my advisor came to me and said, Nathalie, I know that person. He's a geologist, but he's doing this thing also with planet. I think this is a new field. It's called planetary geology. And if you want, I can get you a meeting with him and you can talk to him. And I said, yeah, why not? And I went to it. And this person was at the observatory of Paris-Meudon. And I was 2 days from actually signing on my master thesis in Earth Sciences. I went up to the observatory of Paris-Meudon. I met with that person, which was Professor Cailleux, and I met also with Professor Audouin Dollfus, who was an astronomer. And basically, I left the observatory of Paris-Meudon 9 years later, not with a master in earth sciences, but with a master in planetary sciences and a PhD in planetary scientist, as a planetary scientist. And this is how, for so many years, I had put my dream aside, and I thought I was really going 180 degrees away from it, but really loved what I was doing. So it was not such a hard decision to make. And all of a sudden, at the time where I just decided to let go and follow, whatever path was in front of me, then here it was, back in front of me, right there. So I spent the next 9 years at the Observatory of Meudon and working on Mars and the Viking images. And the focus of my master thesis was on water on Mars. And to get started, because of course I was young in the field and you don't give big studies to do to grad students, I focused on 2 big channels. I was interested by 2 big channels, and one was Dao Vallis and the other one was Ma'adim Vallis. And of course, Ma'adim Vallis was ending up in a very big crater, and the name of that crater was Gusev crater. For whoever knows me, knew that my destiny was sealed that day. I chose that valley. But anyways, a year went by, got my PhD, and started doing a couple of postdocs in Meudon. But there in France, the planetary program was barely started, and there were no position for people like me. So it was really hard. But at the same time, that gave me the time to reflect on the kind of research I was interested in. And I had defended my thesis, my PhD thesis on water on Mars, this time at the global scale. And I was very intrigued by the fact that people were interested in understanding that water flowed on Mars, but people agreed that there were channels, etc. But nobody really cared about where this water was ponding. And when you have water flowing in an atmosphere, it has to pond somewhere. So one of the reasons this happened basically is due to the resolution that we had, or more exactly the lack thereof, because Viking resolution for the topography was really only plus or minus 1 kilometer. So when you want to do hydrological work, this is not exactly the kind of precision you need. So in the end, I was interested in understanding where this water was ponding, and the only way to be sure that we were looking at the depression on Mars was really to pick on impact craters, where you saw impact craters and you saw valley networks converging to them. Then you had a good idea that something could have ponded in those craters. And this is how I started to be interested in impact crater lakes. And for the next few years, I would be pushing the idea that lakes were formed on Mars and formed inside crater. And by that time, the man who would become my husband had joined me in that study. So we're working, the 2 of us, but still I had no job. And I ended up receiving a 9-month postdoc from the French Ministry of Research, and also met somebody who was going to change my life, literally. And that person was Chris McKay. For all of you who are interested in astrobiology and planetary science, you know who Chris is. He is a big figure um, in our field. And Chris was at Meudon at the observatory, and he had a project of a mission, but he didn't have a good landing site. And actually, I was working on Gusev crater, and so trying to understand what it could be as a landing site. So the two of us started to chat, and he said, Natalie, do you want to come to the US and spend your 9 months grant there? And I did it. I just took a suitcase, packed my things for 9 months, and said, you know, if it's the last thing that I do in planetary science, at least I want to give it a shot, because Chris McKay was offering me the opportunity of a lifetime to spend 9 months at NASA Ames. So on Halloween Day, I left Paris with my husband-to-be, and we flew and landed in San Francisco. You can imagine two French guys landing in San Francisco in Halloween. Yeah, the crowd at the airport was interested, and that was pretty much not my first contact with the US, but pretty close to it. So we got settled for 9 months, and I got started. And after 9 months, Chris actually asked me if I wanted to stay. Apparently they liked what I was doing, and so you know, that was either returning to France towards my family and everybody I knew and that I'd been growing up with, but towards an uncertain future, or settling in here where my dream was, everything that I worked for so long, but then meaning that I had to leave everybody and everything behind. And so we stayed. What was a 9-month stay became today close to 21 years in the United States. In the meantime, I became a US citizen because this is a wonderful country, and it really means a lot to me. And then the exploration continued. I started working on impact crater lakes, and I started pushing for the selection of the Gusev crater as a landing site. I started with the Pathfinder mission, but the rover was not right for that at the time. It was just a technology demonstration. And at the same time, since the very first day I arrived at NASA Ames, I knew that as a geologist, I wouldn't be of any use to the exploration community, the planetary community, if I did not understand how robots worked. And so I started to get involved with Mars occult field experiments. I became the deputy of Carol Stoker here at Ames, and really trying to understand how we could solve scientific questions using technology. And this was one of the branches in my job that I developed right there when I arrived at Ames. And on the other hand, I was still interpreting images from Viking and other missions, but I was also starting to get interested in that question of habitability before even astrobiology started. Astrobiology started in 98. I arrived here in 94. And so as time went by, I wanted to understand things from different perspectives. Interpreting images is fine, but if you only do that and you don't have real ground truths to do that, then you can say many different things that can be wrong. And so I decided to do some field work. This is how I started exploring the Andes, those very high lakes, because they have conditions that are very relevant to early lakes on Mars. This is also how I started pushing the testing of rovers and more recently lakelanders for Titan, because I want to understand how we can solve scientific questions with technology. And also how I got myself involved in planetary missions, because Gusev got selected. I had been working for a long time on it, and I became a participating scientist on that mission. So today I continue that scientific path which is to me trying to get a holistic vision on how you solve scientific question. And basically, this is what astrobiology is. It takes the question of life and looks at it from very different perspective. And more recently, I added another branch to my career tree, I'd say, which is the direction of the Carl Sagan Center at the SETI Institute. And I did it because I felt it was important for me to try to share that vision of astrobiology, not only with project teams as I'm doing on the scientific side, but with the scientific community, and try to connect the dots and have the various disciplines that make astrobiology talk to each other. So it's easy when you are talking about earth sciences with space sciences, planetary exploration, etc. But then you have connections that are not obvious yet. And those are the things that are of greatest interest to me. Those are the future connections and the future directions of science that could be of use of astrobiology. So this is what I'm trying to do today by being the director of the Carl Sagan Center, and at the same time still pursuing astrobiology. I am today the PI, the Principal Investigator of the SETI Institute NAI team, the NASA Astrobiology Institute team. And together we are trying to understand how to recognize biosignature on Mars. You all know that there will be a rover on Mars in Mars 2020. Actually, the European already sent successfully a few days ago a probe, ExoMars 2016. And ExoMars 2016 will be making a technology demonstration by landing a little probe in the Schiaparelli crater, but it will also have an orbiter with a TIGO sniffer, which will be looking for methane on Mars. And then in 2018, ExoMars, the big rover, will be sent to Mars. And in 2020, NASA is sending also a mission there. But as we go and start to look for life on Mars, there are a number of fundamental questions that haven't been resolved even here on Earth. The first one is, what is life? What is the signature of life? What are the signature of life? And you have many of those. So we want to recognize what they are. We want to learn how to detect them. And the big question we want to solve as we go to Mars is, what do you sample when you are on Mars? Where do you sample it? And how you do sample it? So these are the questions that with my team we are working on right now. So in a nutshell, that was 23 minutes long. I apologize for being a little lengthy here. This is how I became an astrobiologist and what I'm doing. So I guess I'm going to open the floors to questions now. Well, Graham, I'm seeing your question here, and you're asking what TIGO can offer to the methane issues. And this is a really good thing. Remember, this is going to be an orbiter. Right now, we have MSL, which has been sniffing methane on Mars. And it's not so much that it was the rover has been discovering methane, rather than the fact that this is a variable emission of methane. And if you were expecting this emission of methane to be a background related to decomposition of cosmic material with the Martian regolith, you would expect this to be a background noise and being pretty much not changing over time. So what Curiosity found out is that there is at least one variable source of methane here on Mars. And what TIGO can do will be to look at global scale, not only over the Gale crater, but see if we have variable emissions that are showing up here and there on Mars. Variable emissions are really exciting because they could be related to a geological source, like a still active vent somewhere on Mars. And they also can be related to a biological source. Methane is one of the byproducts of life, and it can be both of them. And we know on Earth, for instance, in volcanic regions, that you would have methane emissions just because you have life near hydrothermal vents. So TIGO can help with that, can help us map this variability of methane on Mars. Now, Shanna, you're asking, aside from your mother's advice to apply yourself to your passion, what other good advice have you given along the way that you would pass on to the up and corners? Well, I'd say never give up. That's the, that's really the thing. And as usual, Mom was right. Apply yourself. But even by applying yourself, sometimes you stumble into walls. And this is not because you did anything wrong. It's just because sometimes circumstances are not right. So, you should apply yourself and continue to push on. But sometimes the wall is here for a good reason as well. It tells you something very precious. It tells you about, who you are and who you are not. And sometimes this wall is just not a wall. It's just a way to redirect you for, to go where you need to be. So the thing that I would say is never give up. Really never give up. And push your dreams. And, you don't want to let the others try to define you. You want to define yourself. You want to define your life. And this is true in science too. We are not always right. And another thing that I would say is that science is not about being right or wrong. And it's not about personal ego. Science is about facts. And you can have hypotheses, and it might be your pet hypothesis. But if it turns out that it is wrong, the right thing to do is to say, this hypothesis has been falsified. This theory has been falsified. And so this is not the right answer. I'm going to look somewhere else. Because if you are not doing this, then you are going to corner yourself in a very, very narrow avenue. And this is not what you want to do. Just like in life, accept that you can be right or wrong, being humble about it, and never give up. Prashansa, regarding the Mars explorers being sent, will they not pollute the surface of Mars with Earth's biomaterial? This is an excellent question. And this question relates to planetary protection and forward contamination. What does that mean? It means that we live on a planet where life really, and finding life is really not the issue. We are looking for life about everywhere else, but here it's all over the place. And at sizes and scales that sometimes we just don't see with the naked eye. And we understand that just putting a robot on a rocket and sending it to another planet, especially Mars, could result in contaminating that planet. And that contamination could be life. So, especially on Mars, because conditions are not that different from the Earth. It was even more true in the past. Right now you have a sterilizing process at the surface, which is the bombardment by cosmic rays and very, very short UV. But still, there are places, if microbe gets into the subsurface, where you have water, they might find energy by interacting with rocks. And who knows, we already know that there are some microbes on Earth that could survive on Mars. And the last thing you want to do when you get to Mars after, applying yourself that much is to find terrestrial life. So how we do that? There are protocol and processes to sterilize our spacecraft. And so we do send spacecraft that have passed all those tests, which is a certain number of micro per volume. And I don't have on top of my head what that number is, but it's very stringent. And this is as good as we can do with the technology we have today. And it's also a very good question when you think in terms of sending robots is already difficult and you have to make sure that they are sterilized. But now we're thinking about sending humans to Mars. And you know what? Humans are excellent for exploration. They will be a lot more efficient. They are more fragile as well, so they will need robots. But in terms of brain power and in understanding what a landscape is about, then they definitely are better than robots. But they have a problem. All of us are microbe factories, and it's going to be extremely difficult to keep contamination in check when humans are there. And I'm always joking that the day we send humans to Mars, we will find life on Mars. And I was not even talking about the astronauts themselves, but in fact, because we are going to bring our microbes with us. For some people, well, for everybody, for the science community, there are ethical questions associated to that. Do we want to do that? Do we want to make sure that life is not, didn't appear on Mars before we contaminate it? You know, these are very, very important questions. And you have planetary protection policies, you have ethic questions. It's something that is looked at very critically. So this is an excellent question, Prashana. Christina, what advice do you have for aspiring scientists who are not good at math? I was one of them. And I became good at math and at physics later on. And, you know, I truly believe that everybody is good at pretty much everything, but we are all wired differently. You have to find a way of learning math that is good for you. If for you it is like for me, for instance, I would learn absolutely anything just by listening to music. For other people, that will be running. Others are taking books and just, you know, eating the whole theories. I'm not good at that, but I'm really hands-on. I can learn anything by listening to music. And if really math is not for you, well, you know, I can tell you something. There are so many fields in astrobiology or in science that don't need that much math at all. So, you know, it's not because you are not good at math that you are a failure. You're a beautiful human being and you have your own skills and you are going to help society in many different ways. So, you know, try many different ways and see with time if you are a practical person. If for you math and understanding theories, etc., is something that is practical. Try that. That worked for me. All right, Bethel, how do I rejuvenate myself? How do you relax your mind? My response to your two questions is first, work, and second, work. Well, you know, I am barely kidding because I have a lot to do these days, but I enjoy very much what I'm doing. And to me, writing a paper is my personal meditation, is my personal yoga. So I can do that. My other way of relaxing my mind is definitely music. I am a music person. I can definitely, you know, just access a state of relaxation that's very helpful, especially when you are dealing with questions of multidisciplinarity and questions about life on Earth or in the universe. It's just that there is a complexity out there that needs for you to have a very clear mind. For me, music does that. And another thing that might make you laugh is driving. And if I drive listening to music for hours, then you can be sure that in the coming days I will be very productive when it comes to papers. So, you know, for other people, different thing. I swim a lot, obviously. For whoever knows me, swimming and diving is probably where, you know, next to music, what is for me the best relaxation method. Sanjoy, you're asking me, well, that was a good segue. Could you tell us what it's like to scuba dive in a lake that's in a crater on top of a volcano? Well, I could sum up by saying it's awesome, but that wouldn't do too much to you. You are referring to my diving into the Licancabur lake in the Andes. I'd say I'm not a religious person, but I'm a very spiritual person, and that was as close as possible as a spiritual experience, which means, you know, some sort of a oneness with the environment. First, you have to imagine, and I think that would be fairly typical of who I am, somebody on top of a dormant volcano, which means, in fact, you know, active anytime, on a scuba diving gear with fins on her feet. So this is basically my attitude in life. But at the moment, with my team, when we entered that lake, we really stepped out of this time on Earth and entered a time machine. we were literally entering Mars 3.5 billion years ago, because this lake is so high that the environmental conditions out there are pretty much like what they are on Mars. So for us, just looking and diving in this lake was like being in a time machine and seeing Mars in the past 4 billion years ago. And then there was the other aspect, which is more of a physical aspect. We were in very cold water. We had our dry suits and a lot of weight on Earth, which for me was really a challenge, because first, I'm not that big and I have about more than half my weight in lead around my waist. And I felt like a whale, and even in the water I could feel it. I am a free diver normally, so I wouldn't go with that much equipment. But there we were. And the Cabrol Lake is completely transparent. So you have to imagine Arctic blue waters where each ray of the sun is being diffracted from the surface down to the bottom. You can see every single one of them. And I surprised myself to just being suspended there and looking around me, and just totally forgot about time and where I was. And then, of course, you have the mission, and you have the samples, and you have all of that, and you have a tank of air that you need to think about as well. But I started to joke with myself. I felt so comfortable and so happy out there that I was thinking to myself that if this volcano erupts now, well, I might have a chance. I might be the first scuba diver in orbit, and I have enough air that maybe I can wait for the space shuttle to pick me up. So it was an amazing experience by all means. And these are the kind of privilege that, living your passion every single day can bring you. So if I have one piece of advice to all of you, whatever your passion is, go for it. Never let go, because every time you wake up in the morning, you will not go to work. You will live your passion, and there is a big difference for that. All right, Bethel, if you were to design a microbe to survive on Mars, how would you want it to behave? Microbes misbehave all the time. They are getting us sick. But to respond to your question, are there genes, genomes that we can learn from or study or even improve in order to design more resilient microbes that can survive on Mars? So to respond to your question, Bethel, there are actually currently microbes we know would survive on Mars. And this is the issue, going back to the previous question about cross-contamination and making sure that we are sterilizing rovers. But if we were now to on that stage where we have colonized Mars, that we know for a fact that there is or there is not life on Mars, and we want to, say, terraform Mars and use some form of life to do that, how would we do that? Well, you would first want to have microbes that are highly resistant to radiation. And I think that the one we have on Earth, and especially the one we saw in the Andes, are a good start to that. But you might want to engineer them to resist what we don't have on Earth, is a very short UV, which means UVC and other nasty cosmic radiation. And what you want to do as well is to make sure that, at least at the very beginning, you would be growing whatever you want to grow on a shelter, on a greenhouse, and that you are starting an atmosphere that is substantial enough that then you can expose those microbes and those bacteria so that they can take the gas from the atmosphere and do what life did on Earth 3.5 billion years ago, which is to extract CO2 and pump oxygen. So depends on what you want to do with your microbes. If you want to biomine, some people are using bacteria to actually leach minerals and extract the good stuff that you can use for the industry. So that's different. You'll probably do this in caves or in mining places on Mars, but that's really much farther along. At this point in time, we're thinking in terms of what kind of life would be useful to help us terraform Mars, and what kind of life could help us pump some good gases into the atmosphere of Mars. So these are the kind of thing, but bioengineering will certainly be an important thing. And, you know, I am not talking about genes or genomes, etc. I am not a biologist. I'm a geologist by training. So you would need to ask this question to somebody whose expertise is really microbiology. Graham, if you had to bet on it, what do you think the next major discovery in astrobiology might be? Well, let's put it that way. I think that our generation is the best position to find life in the solar system. And whether it's going to be on Mars or it's going to be on one of the outer solar system moons, I don't know, but it's going to happen. I think that we have proven to ourselves that our thought process is the right one. For Mars, for instance, we developed an integrated program that first looked to understand what the geology of Mars was throughout history. Then we started to understand what the climate was throughout history. The first mission was Mars Global Surveyor, the second one was Mars Odyssey. And then when we saw that, we put two and two together and say, well, Mars must have been habitable in the past. And so where would that be? Well, in places where you are seeing ancient traces of water, whether it's morphology or mineralogy. And then we sent the Mars exploration rovers, and guess what? Both of them found what they were looking for. And they demonstrated that Mars was habitable for life as we know it 4 billion years ago. And MSL landed, and MSL did find an even more extraordinary site, where we're seeing the complexity of habitability in front of us. So we're thinking along the right lines here. And the next step is to search for life on Mars. So Mars was habitable for life as we know it. Conditions were not that different. So there is a good chance that we might find what we're looking for with Mars 2020. If not Mars 2020, we'll find it because I'm pretty sure that something happened on Mars. And to me, the most exciting part is not necessarily that we could find life on Mars. That would be tremendously exciting and very important too. You all heard about these different hypotheses about the life developing on Mars. One of them would be that Mars developed its own life, and that would be exciting on its own right because that would be to Genesis in a planetary system of 8. So this is incredible what it tells us in terms of the ability of life to develop. But to me, what would be an even more exciting thing would be that actually Mars and the Earth could share some of the seeding material that led to life on Earth. And this would be a possibility because when they were formed, Mars and the Earth exchanged a lot of material through impact ejecta. And they traveled through space, and pieces of Mars landed on the Earth, and pieces of the Earth landed on Mars. And it might be that the two planets have been seeded by the same material. Why is that important? First, well, we would be cousins. That's a good thing to have family nearby. But second, it would give us the clue that we have been looking for so long here on Earth, and that we will never find here on Earth is where are we coming from? How this transition before prebiotic chemistry to biology happened. What is it? We don't know on Earth, and we will never know because the Earth is an active planet. It has plate tectonic, it has erosion, and all the oldest rocks have been totally erased. The first traces of life we are finding, which might be indirect traces of life, are going back 4.1 billion years ago on Earth. And this is pretty much as old as you can get in terms of rocks. And life was already there. What we don't have is the answer as to what is the transition between prebiotic chemistry and life. And if by any chance we're sharing seeding material with Mars, and Mars developed on, and life developed on Mars, then it may be that somewhere on an outcrop on Mars lies the answer to our question. Who are we? Where are we coming from? And how it is that prebiotic chemistry became life. So to me, this is important. Whether we're going to find life first on Mars or on Europa or Enceladus, doesn't matter so much. I think this is something that our generation is going to achieve. As far as Europa and Enceladus are concerned, the excitement is different because if the the celestial mechanics works well for Mars and the Earth to exchange material. It's a lot more complicated with the other solar system, which means that if we are finding life on Europa or Enceladus, it has to be different life from what we have on Earth. So a second Genesis or even a third Genesis, who knows? So lots of exciting stuff ahead of us right now. Svet, can you speak a little about your work-life family balance, especially as a woman in science and as someone with family in another country? And P.S., my husband is French too. Well, you know, it's very specific in my case, in that unfortunately I don't have very much family left, and my family really is my mom and my husband. And so my mom is in France, and I have her come here every winter. We keep her with us because winter is colder, as you know, in France. And so it's a good period of time. We have her with us for 3 months here in California. So we enjoy each other. But the rest of the year, yeah, it's a long distance relationship, and it's not an easy one. My mom has always been my best friend. And so it's a lot of sacrifices, but you know, when I look back, basically she did what every mom does, which is sacrifice her own life first to give me the means of doing my studies and achieving my dream. And today she's also sacrificing herself and my presence near her to make sure that I can achieve what I want to achieve. So, of course, there is the possibility that she will come here and live with us. I offered that to her, but for now she's happy in France with her friends, etc. So as long as this is true and she's happy, I'm happy too. But this has required some adjustment. I personally don't have children of my own. My husband had 2, and so he sees them from time to time. We see them from time to time, a little less now because they are grown up and living their own life, which is good. But the way I balance my life with my husband, it's pretty much a very bad example because my husband works with me. He does exactly the same thing. We're in the same office and we are 24/7 together. Many people are asking me, how can you survive like that? And my answer is that, you know what? We love each other and we don't even ask. The only question I have is that, how would I do that, you know, if it wasn't the way it is? So I'm blessed, privileged, and it's also, yeah, certainly, you know, easier when you are sharing not only your life with your husband at home, but also at work. And we are going along very, very much. We are joined at the hip, literally, and often I say we are only, you know, the same soul in 2 different bodies. So that helps. That helps. But there are so many other people in this community who don't have the same chance I have, and definitely need to juggle being a mom, being a wife, and being a scientist and an active woman. And well, that means that we have 3 jobs, and each of these jobs is a full-time job. So I understand that, definitely. Richard, I'm going down here the list. Assuming that rocks have been transported between Earth and Mars and that microbial life is found on Mars, what is the most probable age difference? Let's see. You know, I'd say that happened as both planets were forming, which means that rocks coming from both planets were incorporated at the time of the heaviest bombardment, because this is at the time where you have the biggest asteroids and comets and pieces of rock flying into the solar system, which means that the ones that had the most energy to allow a transfer through ejection to both planets. So I would say that, you know, those 2 planets were seeded at about the same time. And interestingly enough, they had conditions that were very much alike at the same time. And if you're looking back at the development of life on Earth, the first fossil we have are 3.6 billion years ago, 3.5 billion years ago, and these are the stromatolites in Pilbara in Australia. So 3.5 billion years ago is also a very interesting date on Mars, which is at the time where climate change on Mars, and everything was transformed. Mars lost its atmosphere, lost its water, and was bombarded by UV. But that means that, Mars still had the same time as Earth to develop at least simple life. And when you are looking at the Pilbara samples in the stromatolites, these are already cyanobacteria. So it's simple, but it's not that simple. And to me, there is a good chance that if anything happened on Mars, it happened in the same time frame. Now, you're asking if there are plans for sequencing Martian DNA to determine last date of transfer. Could this be done remotely? You are entering a realm that is not too familiar to me. This is not my expertise. The only thing I could say, and this is a very high level response here, is that if we're related with Martian life, we probably share some DNA. And that would be very interesting to try to track back. But also what you know is that life adapts very much to its environment, and the environment sort of, I wouldn't say adapt, but reacts in function of the life that it's supporting. This is what we call the coevolution of life and environment. Basically what it says is that you have prebiotic chemistry, and then the conditions are good for life to appear, life appears, and all of a sudden from the very beginning start to mess up the environment. This is what cyanobacteria did. The terrestrial atmosphere was not what it is now, and it's just because you had this tiny blue and green algae pumping oxygen into the atmosphere that you breathe the air you breathe today. In doing so, they actually killed 99% of life on Earth at that time. So we definitely, can see that Martian life, even if it had the same starting point as the Earth, would have been interacting with its environment in very different ways, because the environment on Mars is different. So what is this coevolution on Mars? What is this interaction? I don't know. But certainly you would see that evolution in the genes. So I don't have much more of an answer now, but definitely we would see that evolution. Determining the dates of transfer, I don't know, people today are finding genes in modern humans that you can trace back to some of the early human species. So we are now trying to trace Neanderthal in Homo sapiens. So, yes, probably, but to have a better answer, you need to ask somebody who was doing genomics. Kai, what are some limitations technology has when it comes to sampling and analyzing samples collected via rover? And how do you test them in the field? It's an excellent question. And that goes back to the scientific process, which is that before you put a payload together, you have to articulate a science question and really define what you are looking for. And the limitation that I see now, we have a pretty good handle on trying to understand what the mineralogy of a rock is, what the composition of a rock is. With Raman, we'll be able to do organics as well. And there are a number of things that we know how to do well. The limitations we do have that would require at this point in time a sample return would be to be able to slice rocks in the field, or to date rocks in the field, for instance. Because if you could have this answer right there, then you could interrogate your outcrop or your samples in different ways. Whereas right now, you would have to bring those samples back here on Earth and put them through the labs. Or you will have to wait to have humans on Mars and wait for them to bring their labs with them over there. So the other obstacles that we do have using rovers and robots in general, first is the time delay. Obviously, when you command a rover, you can enter a sequence and then your rover will execute that sequence, but you still have to wait, for the communication to happen. So there is time that is being wasted doing this. And then, you have to wait and sit for the experiments to go on. And since the rover is the only one there, it's not that he can do something else much while he's doing an experiment that requires to be sitting there still. So there are constraints, but if you actually compare what the constraints are, using a robot on Mars or on any other planet, and not having anything at all and just trying to think about, what we could do until we can send humans, I think we have a pretty good deal right now. We have been able to achieve a lot. All right, Jacob, you're asking me what are my favorite styles of music. Oh, absolutely everything. Absolutely everything. As long as it pleases my ear, I can go from classical music to rap to rock to pop music. It's just that it had to catch my ears, and I have to like it. I am no style specific. Certainly, I would say that classical music is definitely the best to relax and expand your mind. Modern music, rocks, etc., takes you in different places, but it's a lot more energetic and maybe less favorable to let your mind wander in those places you want to actually address some concepts. But definitely all styles of music. I can be humming on everything. I am a music junkie. I really do. So as I said, driving along the coast here in California, which is a blessing, listening to music is as good as it gets. Parashansa, we are searching for life with a geocentric notion. Is it foolish to expect life not based on carbon as on Earth? No, absolutely not. And you raise an excellent point, is that today, and that's true for both astrobiology across the roadmap, but also a very specific aspect of astrobiology, which is the SETI aspect, which means, advanced life. In all the spectrum, we are looking for life as we know it. And this is a huge limitation. But at the same time, this is an excellent starting point. You want to start with something you know. And you know why it's good to start with that? Just because we know on Earth, at least, it's a model that works. We know that in some condition, physics, chemistry leads to biology. So should we limit ourselves to that? Absolutely not. And especially when it comes to ET and exoplanets and etc. I think that, there is a good deal of the answer in why we haven't heard back from ET, because we're just searching for another version of ourselves with a very limited technology so far. I don't think that ET is dead, or that, ET doesn't want to talk, or we are in a zoo, or, I just think that we should be much more humble than that. We're just starting and we are lacking imagination. And imagination is very, very important when it comes to science. I'm not the first one to say it. Albert Einstein was, definitely a proponent to the fact that imagination should drive our thought process. But what we need to understand, we need to have a very good handle onto what is life as we know it, because then we can define life as we don't know it as one minus what we know. And then I think we would have to let go of intellectual walls. So I really like your question, because this is very much an avenue that I'm exploring right now for the SETI Institute, as to how do we think of life as we don't know it. Obviously, this is probably the broadest and largest field in astrobiology, because when you look at the evolutionary continuum as what it took for us to be here, well, life as we don't know it is probably the rule in the universe and not the exception. But today we start humble by, like children at school, learning the basics of our own alphabet. And when we learn the letters, we'll know how to mix them and to understand how many words, the words being the many type of life that the universe can have. So definitely an excellent question. Christina, does planetary geology involve a lot of math? That's what my second goal next to astrobiology. Well, planetary geology will lead you to environment. And geology, some of it involves some math when you're dealing with structural faulting, when you're dealing with plate tectonics, etc. But there are areas of geology that don't require that much math at all. So, depends. And I think that your second goal next to astrobiology, I think you can fuse that together, because geology is one of the fields of astrobiology. So go for it. Go for it. And pick the areas where you feel you are good at and you can give the best of yourself. But don't be afraid by math. You will understand them. It's I will say here what a professor of geology told me one day is that, I can teach you as much as I can in the classroom, but then, I can give you classes for hours and days and weeks, and I'll take you in the field, and all of a sudden you will learn all the exceptions to what's in the book. And pretty much you will discover as life goes that nature is only one big exception. So, math is only the beginning. Do what we can with math. There is not so much in planetary geology, and go for it. Run with it. Prashanta, you got rejected by all the graduate school you applied to due to above average marks. Above average? I wish to do a doctorate in exoplanetary science. What advice do you have for me? Then I need to understand if it's above average or below average. If it's below average, then, continue to learn and find people who can teach you in ways you can understand. To me, everybody can learn and can learn pretty much everything. It's not a matter of a person not being intelligent enough. It is to the teacher to find a way to teach to a person. So this is one thing. And, get a good solid background. Take your time. It's not a sprint. Life is a marathon. Doesn't matter if you graduate when you are 25 or if you are 35, as long as you fulfill your lifelong dream. So, continue to learn, find the right people to teach you, and continue to apply yourself. This is the advice I can give you. Shana, and I see that it's pretty much time, but I will take the last two questions here, and then we will wrap up. Tatiana, since astrobiology is such an interdisciplinary science, is it an advantage to specialize in multiple fields of science like geology alongside microbiology? Another very good point. As I said, I am not a microbiologist. I am a geologist and a planetary geologist by training. I also have an earth science background. So I did minors in earth science, major in planetary geology. And I don't look that stupid anymore when, I need to discuss a little bit of microbiology, because I watch my colleagues coming with me in the field who are microbiologists and started to teach me a little bit. So you can find the places where you are good and find major and minors that can complement each other. I would definitely suggest that you take some classes either in geology and, on both. You take classes in geology and microbiology, and also something that is extremely useful for all the scientists today. A couple of things, how to deal with data. Data processing is extremely important, because this is structuring your mind and how you analyze your data. And also science writing, very, very important. All of you who want to be scientists, you will need to write proposals. You will need to convey your ideas, and you will need to make sure that reviewers understand what you are saying. So don't be afraid to go back to school and learn how to write science papers. So this is the kind of advice I would give you. Chris, in order to learn what conditions are necessary for life and to inform astrobiologists as to where to look for life, it seems natural to set up conditions in the lab to get non-life to become life. Miller did that in the experiment a long time ago. And can you speak on the relationship between artificial life and astrobiology research? So, AI, artificial intelligence, is, remember, as far as I can tell now, although there are programs that are a little bit scary, artificial intelligence is still being created by non-artificial intelligence. So right now, artificial intelligence is a way for us to find ways of perceiving the environment in different ways, and to learn how mathematics and different probabilistic ways of perceiving the environment could help us interact in different way with that environment. So artificial intelligence also help us to extract information that our brain don't have the power, the mathematical and processing power to find in a timely fashion. So artificial intelligence for me, I use it when I'm doing rover field experiment, or the Lakelanders experiment was a good example of that. I wanted to make that robot really a teammate and not only an extension of a team, which means that when you are on Earth and you have a rover on Mars, say, you write some codes, you send your command, the rover executes, has to wait for the orbiter to come, or has a direct to Earth and transmits the data, it comes back, etc., etc. What I wanted to, because the Lakelanders was a project that was thinking in terms of Titan and landing Lakelanders on Titan, then you have a real serious communication problem, not only a communication problem because it's long, but also an energy problem. Basically, you have a little light bulb that's communicating very little data. So if you are practicing planetary exploration the same way you do on Mars, you are doomed. You are doomed to a very low productivity mission. So what I wanted to do was to give some smart to my robot so that it would understand what nominal environmental conditions are and what would deviate from those normal conditions that would be of interest to the science and its mission. So we did that and we demonstrated that, and there are so many applications. So artificial life and artificial intelligence can be extremely useful for us. Artificial life as in, you know, creating new forms of life that can develop on their own. I don't know about that. You have to talk to biocomputing people. But definitely, I think that it can support astrobiology research by helping us understand what is the probabilistic nature of life evolution and development. For me, that would be the greatest interest I would see in that. Anyways, we are now over an hour, 7 minutes. I think that Sanj might want to wrap up this chat. It's been a pleasure, and I'm around, so maybe we'll have a chance another time to meet again and go for more questions. Thank you all.